All right, so we're going to start with a water potential problem. And remember that water moves which way in terms of water potential? High water potential to low water potential. Now, if you look at this problem, sides A and B are separated by a semi-permeable membrane. The triangles represent solutes that are dissolved in water. If the volumes of the two sides are initially equal, so we're saying that there's the same amount of fluid in here, and the membrane is impermeable to the solute, which way will water flow? Well, at first glance, which way should the water go, you think? You think it ought to go this way, right? Because water tends to move from where there's less solute to high solute, right? But I'm going to tell you that's not the right answer, even though at first it looks like it is. Because what added variable do we have going on here? Pressure. And that's why water potential is important. It doesn't just take into account solutes, it also takes into account pressure. So the pressure is pushing out in all directions, right? That's the way pressure pushes. It pushes outward in all directions from where the pressure is higher to where it's lower. So the first thing that we're going to have to do is calculate the water potential. And that's what the next part asks us to do. And you guys remember, I hope, that to calculate water potential, it's solute potential plus pressure potential. So for this first problem, for side A, we've got negative 1.7 and positive 2. So to calculate the water potential over here, it's just 0.3, right? You add them up, positive 0.3. And then on the other side, 0.6, negative 0.6 plus 0. The water potential over here is negative 0.6. And again, water moves which way? High water potential to low water potential. So the water is actually going to go this way because of what? The pressure. Even though in terms of solutes it wants to go this way, because of the pressure pushing outward on it, the water actually goes that way. So the rest of the question here, which side is hypertonic? This side, because hypertonic means more what? More higher solute concentration. This side would be hypotonic. And normally water moves from hypo to hyper. But in this case, the pressure is not letting that happen. And then finally, what will happen to the volume of side A? It's going to go down because water's moving over that way. And that means the volume over here is going to go up. Does that make sense? Let's switch over. Switch over and do a surface area to volume. This one will be really easy. Listen, guys. So, on this problem, we've got a rectangular solid. We've got the length, the width, and the height. And we want to find the surface area, the volume, and the surface area to volume ratio. So if we draw that out real quick. So here's our rectangular solid. It has a length of 6. It has a width of 4. And it has a height of 3. So if we're going to find the surface area, we got to find the area of each side, right? So let's just look at the front. What's the area of the front? Just this front side, what's the area? 18, right? 8 times width. And we got a front and we got a back. So we got 18 times 2. 36. All right, then let's get the top and the bottom. What's the area of the top and the bottom? 24, right? 6 times 4. So we got 24 times 2. And then the sides. What are the area of each, the left and the right side? 12, right? 4 times 3. So we got 12 times 2, 24. So to get the surface area, just add those up. So 36, 24, those add up to be 60, plus 48, 108. And it would be 108 what in this problem? What kind of units? Millimeters, square, area, always square units. All right, so that's the surface area, the total surface area. To find the volume, when you have a rectangular solid, what do? Length times width times height. That's in your formula sheet if you don't know that. Length times width times height. So 6 times 4 times 3 be 24 times 3, which is what, 72? And that would be what units? Millimeters cubed. So our ratio, surface area to volume, is 108, the surface area, to 72. We don't normally write it that way, though. How do we normally write it? Normally you go 108 divided by 72, right? Go ahead and divide it out. And you're going to write it this way. 1.5, which is what 108 divided by 72 is, to 1. Is that a good surface area to volume ratio for a cell? You want that ratio to be very, very big, right? That's probably not good. Which means this cell is probably not very good at what? Diffusion and osmosis and those kinds of things. So the next part of this problem is to take a little bit smaller cell. We'll do the same thing with it. So let's take away the 6, take away the 4, and the 3. Put back in my line. On the second part of the problem, now the cell's 4 long, 3 wide, and 2 tall. And again, we do the same kind of calculation. Surface area. What's the top and the bottom? Top and the bottom, what are their areas? 12. 4 times 3. So if we got 12 times 2, top and bottom. Front and back, what are their areas? 8. 4 times 2, right? That's this side. 8 times 2. We've got a front and a back. And then the left and the right, what are their areas? 6. So that's 6 times 2, 12. So if we add that up to get the total surface area, that would be 40 plus 12, 52. 52 millimeters squared. And to find the volume, again, it's length times width times height. So 4 times 3 times 2 would be 24, would be the volume. Which gives us a surface area to volume ratio of 52 to 24. Now, like I said, we usually are not going to leave it that way. We're going to go ahead and go 52 divided by 24. And that gives us 2.2 to 1. So because it's a smaller cell, it has a what? Higher surface area to volume ratio, and therefore it's better at what? Diffusion and osmosis and everything in between. Everybody okay with that? All right, let's go a little further. Here's something similar, and I'm gonna blow through this one pretty fast. Same kind of thing, except this time we're not looking at a rectangular cell, we're looking at a sphere-shaped cell. And you may not know the formulas for these, but if you grab out your formula sheet and you look at the back, it gives you the formula for the surface area of a sphere, and it also gives you the formula for the volume of a sphere. Those are on the back left column, bottom left of the back. So it tells you that to find the surface area for a sphere, got to use 4 times pi times r squared, r cubed. All right, so, and not cubed, that's squared. We'll start over with that, guys. So 4 pi r squared. So in this first cell, the radius is 3 millimeters. So our surface area is 4 pi times 3 squared. I'm just going to go ahead and write 9 there. 3 squared. So we multiply that out and we get 36 pi. I would go ahead and multiply out the pi. So 36 times pi times 3.14. We get about 113. Yeah.
So on the test, they're going to ask us to use pi and all. Would you rather do it less like, well, I'm kind of a stupid question now that I think about it because we're
Right, just 3.14.
Okay.
You're not going to have the pi button on your calculator. Yeah, because you're using the cheapy calculators. All right, so we get a surface area of 113, and for the volume of a sphere is 4/3 times pi r cubed. So in this case, you're going to go V equals 4/3 pi times 3 cubed. What's 3 cubed? What's 3 cubed?
27.
27. So now we multiply that out. 27 times 4 divided by 3 times 3.14. And we get, kind of strange, we get 113 again. Which means our surface area to volume ratio is what? 1. 1 to 1. Is this cell going to be good at diffusion or osmosis? It is not. I'm not going to take the time to work the second one of those because it's just plug it in and do the same exact thing. You've got the formula for the surface area of the sphere. You've got the formula for the volume of the sphere. Calculate it out. And remember that bigger is better in terms of surface area to volume ratio. Smaller is better in terms of what? It's the actual size of the cell, right? Smaller cells are better because they have a bigger surface area to volume ratio. All right, let's go to something a little different here. And again, an easy one, mean median mode. That's one of those data things that they're going to put on there. And also range. I'm not sure that we talked about range. How do you find the range of something? Yeah, the biggest minus the smallest. It's just kind of a way to look at the differences in your data. So for this one right here, the range for number 1, what would the range be? 5. The difference between the biggest number and the smallest number. So we've got a range of 5. Remember to find well, let's go ahead and find the median. We can find that one really easily. What's the most common number in our, or actually the mode. Let's find the mode first. What's the most common number in our set? 5. So that's the mode. To find the median, we need to put them all in order from smallest to biggest. So we've got 4 5s. Then we've got a 6. We've got a 7. We've got 2 8s. And we've got a 10. So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9 numbers. Which means our middle number is going to be the 5th one, right? So the 6. Remember that if we had an even number, what do we have to do to get the median? Yeah, you would have, let's say that these two were both in the middle. You'd have to average them, right? But in this case, we don't. So our median is 6. And then to find our mean, we just add them all up and divide by 9, the total number of entries. So let's see, we've got we've got 6.6 as our mean. I don't think any of that is new for you. Just remember the range is the difference between your biggest and smallest number. The median is your middle number, if you put them in order from smallest to biggest. The mode is your most common number in your set. And the mean, you know how to calculate the mean. The mean is the average. And we haven't done a Q10 problem in a long time. So let's look at this Q10 problem. Q10 is a way to calculate how dependent a reaction is on heat. The bigger your Q10 value, what does that mean about that reaction? Well, it means the more it needs heat. What kind of animals is this going to be an important calculation for? Try again. Ectothermic, because the temperature inside an endothermic animal should what? It should be constant, essentially constant. But in an ectothermic thing, your metabolic rate changes with what? With your environment, with temperature. So it's more of an ectothermic kind of thing. So let me go to a blank slide where we have a little space. If you look at your formula sheet, you have a formula for Q10. And it is, now sometimes you'll see this written as K, sometimes as R. What do the K and the R stand for? The rates. And then we take that rate and we raise it to this power. Now remember that the 2 and the 2, those have to be together. This temperature is when that rate happens. This temperature is when this rate happens. So if we look back at our problem, I've got it here on the sheet. An experiment to measure the rate of hydrogen peroxide decomposition by catalase at 2 different temperatures was performed and yielded the data shown below. Calculate the Q10 to the nearest hundredth. So our highest rate was 0.0924. And that was at 25 degrees Celsius. You're always going to put the highest rate up here for K2. So 0.0924. That's over the lowest rate, which in this problem was 0.0763. The units don't matter so much here because what's going to happen to them? They're going to cancel. And then we want to raise this to the 10 over the highest temperature, which was 25, minus the lowest temperature, which was 15. So essentially, does the power even matter on this problem? Because it's going to be 10 over 10, right? It's going to be 1. So all we do to get our Q10 is divide out 0.0924 by 0.0763. So we end up with a Q10 of about 1.2. Q10s are usually going to be between like 1 and 3. So this particular reaction is probably not too dependent on what? Probably not too dependent on heat. Remember, the higher this number gets, the more dependent on heat the reaction is. And it's mostly just important in ectothermic kinds of things, where temperature varies. What are some ectothermic groups of organisms? Reptiles, amphibians, not birds, ectothermic. Fish, keep going. Insects, most all the invertebrates, right? The 2 groups of endothermic things are mammals and birds. All right, let's go to the next one. This is one that you saw in the last study session. And it's one that we really haven't done much in class. But it's an easy thing to do. Let's read through the problem first. Scientists use various different methods to try and establish the age of fossils. One such method for fossils less than 75,000 years old is to compare the amount of carbon-14 to normal carbon-12. Both forms of carbon are assimilated in the organism as it feeds over time. Are you guys radioactive?
Yeah.
Yeah, because you have some radioactive carbon-14 in you. It's everywhere. You're not very radioactive, but you're a little bit. When you die, you stop taking in what? Carbon. So what happens to the level of radiation in your body the longer you're dead? It goes down. Because carbon-14, as it decays, as it goes through radiation, what does it become? It becomes nitrogen, which isn't what? Radioactive. So the amount of radiation in you the longer you're dead goes down. The amount of radiation you're giving off. And it turns out it does that in a fairly predictable rate. When we're talking about radiation, we talk about this idea of half-lives. The half-life of something is the amount of time it takes for about half of it to what? Decay, to go away, to become something else. This is radiocarbon dating. Yep. And for carbon-14, that half-life is about 5,730 years. About every 5,730 years, every half-life, about half of the amount of carbon that you started with goes away. One half every 5,700 and something years. So what this wants to know, if the half-life of carbon-14 is about 5,730 years, then a fossil that has 1/8 the normal proportion of carbon-14 to carbon-12 is probably about how many years old. The easiest way to do this is to set up a little chart. And let's do it this way. Let's do time and fraction remaining. I'll just say C14 left. So at time 0, when you're first starting, it's all there, right? So we'll say 1 or 100%. But after one half-life, how much is left? 1/2. Let's do it in fractions. 1/2, right? After one half-life, a half of it's left. After two half-lives, how much is left? A half of a half, which is a fourth, right? Half of a half. And after three half-lives, a half of a fourth, right? Which would be an eighth. And that's what the problem tells us we're looking for, right? Is how long is it until only 1/8 is left? Well, it's three half-lives. But it wants that in terms of years. So we're going to take 3 times what?
5730.
3 times 5730. And that is 17,190 years old. So that's a brief description of how they use radioactive carbon dating, carbon-14 dating. Yep, long time, but that's easy math, right? Not difficult at all. Just remember that every half-life, you lose half. Just do it in a chart like this, and it's easy to figure out.
Does it always tell you that it's 1/8 or whatever the number is?
It could give you the number of years instead. I mean, it could say it's, it's been, well, they could give you a number of grams. They could say you started with 20 grams, and now you only have 5 left. How many half-lives did it take to get there? Well, 20 to 10 would be one half-life, and 10 to 5 would be another one, right? So it'd take 2.
Wait.
If you started with 20 grams of carbon-14, and you only have 5 left after a certain amount of time, and it wants to know how much time. 20 to 10 would be the first half-life. 10 to 5 would be the second. So it takes 2 half-lives.
Yeah, okay.
Does that make sense? Like I said, the math on this there's none of this math that's hard. It's all easy math. This problem, I'm going to save until class tomorrow and Thursday, because this is a topic we're going to go over then. So I'm not going to do this one today. Let's do a Hardy-Weinberg. Actually, since we just did Hardy-Weinbergs, let's go to something easy first. Let's do these two at the back first, and then we'll come back and do the Hardy-Weinbergs and chi-squares if we have some time. All right, this one right here, the probability question. There's pretty much always going to be one like this on the test. And remember, we want to do it one set of genes at a time. So first of all, we look, what are the chances that this right here and this right here make that right there? I heard two different things. Which one's right? It's 1/4. Now, if you can't visualize that, draw the Punnett square between these two right here. So we got big A, little a, and big A, little a. And I'm hoping that you can see that the only one that turns out big A, would be that square right there, right? So it's 1/4, 1 out of 4. Then we look, what are the chances that that and that make that? And that's 1/2. And again, it'd be the same square with B's instead of A's. This would be 1, and so would that. So 1/2. Remember, we multiply these probabilities. And then finally, the chances that that little c and the big C little c make a big C little c offspring. I'm hearing different things again. How about 1/2? And again, if you can't visualize it, the little this offspring right here can only make little c gametes. This one can make big C's or little c's. So I'm hoping that you see that this would be big C, little c, and this would be little c, little c. 1 out of 2. So our answer is what? 1/16. Multiply them out. Jake, you okay with that now?
Very good.
All right, those are really easy ones. Don't make those hard. And the same can be said for this next thing. This next thing, we're doing what's called gene mapping. And we're doing the mapping based on what? How often two genes cross over. And the important thing to understand is that the closer together two genes are on a chromosome, what's going to happen? The closer they are, they're going to cross over less. The further apart, they're going to cross over more. By the way, what do we call genes that are on the same chromosome? Somebody said linked. Linked. Gene linked. So what we take here, I like to start with the ones that seem to be farthest apart on the chromosome, which according to our data looks like it's going to be what? A and B. And write those at opposite ends. A and B. And I'm going to go ahead and start mapping it out here. 75% crossover, or what unit can we call that sometimes? A map unit. All right, then I'm going to try to figure out what's kind of close to A. And the one that's kind of close to A is C, right? C could go right here, or C could go right here. Let's see if we can figure out which place it goes. And the easiest way to do that, we know that gene D is only 40 units from A. So D could be over here somewhere, or D could be over here somewhere. If D's over here, 40 units from A, the furthest C could be from D would be how far? If D was over here, and this distance here was 40, what's the A to C distance? 15. So that means that the C to D distance would have to be what? 25. And that's not it, right? So that's telling us, well, one other way to look at it though, C could be here, right? And if C was here, and that distance is 15, and D was here, and that distance is 40, then this works, right? C to D being 55, that works. So let's see if the rest of it works. If D's here and B's here, does that work? That doesn't work, right? So what we've got to do then is move two of these around. C's going to have to be here. And that distance is 15. D's going to have to be in here. This distance is 40 map units. This still works, right? The C to D distance is still what? 55. That works. If this is 40 between A and D, and the distance between A and B is 75, that means the distance between D and B has to be what? 35, and that's what it says. So that's the only way it'll work. So you would write down for your answer CADB. Or remember, they might also flip it and write it the other way, BDAC. Because are we really determining the direction here? We're just determining the relative order that they're in, is all we're doing. That's what mapping does. Make sense? All right, let's do a slope problem, a rate problem. Find the rates. Now, first of all, this is a graph of an enzyme reaction. When is the rate of an enzyme catalyzed reaction always going to be the highest? Okay. At the beginning. Because why? There's more substrate, right? The concentration of the substrate's higher, so it's easier for the enzyme to what? To find it and bind to it and work with it. And notice that this graph is showing us that the longer time goes, what starts to happen to our slope? It starts to decrease and become more and more flat. What's happened up here by the time we get up here, what's happened with our reaction? It's stopped. Because what's the rate of what's the slope of a horizontal line? 0. The reaction has stopped at this point. And it stopped because what happened? The substrate's gone. There's no more left. So now let's calculate a little rate. Find the rate between 10 seconds and 20 seconds. So first of all, we have to determine points. At 10, our y is what, maybe 7? So 10 and 7, that's 1 point. And 20, at 20, our y is maybe 13 or 14. Let's say 14. Find rate, you find slope. Change in y. So 14 minus 7. Over, change in x. 20 minus 10. You have to do y over x. So we end up with 7 over 10. Or, go ahead and divide it out. 0.7. 0.7 what? Moles over seconds, right? Moles per second. And then the other part of this, let's go ahead and find the rate between 30 and 40. So at 30, we're at maybe 16 or 17?
16.
Like 16, okay. And at 40, we're at probably more like 18-ish. So change in y, 18 minus 16, over change in x, 40 minus 30. So we're at 2 over 10, or 0.2 moles per second. And we can see that the rate is going down pretty quickly as we go longer in terms of time. Good with that? All right, let's back up and work one of these chi-square or Hardy. Y'all rather work at chi-square or Hardy-Weinberg? Chi-square. All right, let's try this one. Everybody take a minute and read this problem first of all. So we've got some flies, some fruit flies. We're crossing a female with white eyes with a wild type male. All of the F1 males have white eyes and all the F1 females have red eyes. As soon as we see that, what do we know? It's X-linked. Males and females are affected differently. Now, in the F2, it kind of evens out, but we'll be able to explain that. So I'm going to go to a slide where I have a little space to write. Now it's telling us that we're starting with a female with what in our P generation, the female X, X, and the male X, Y. Remember, if it's X-linked, you have to show the X and Y chromosomes. The female has white eyes and the male has red. And then in the F1, in the F1, all the males have white eyes and all the females have red. We need to figure out which one's dominant. She started off with white eyes. These all got white eyes. Does that make sense? If she being white and her son being white, does that make sense?
Yes.
Yes, she's got the only X chromosome he gets, right? But this female over here, she's got red eyes. She got an X from mom and an X from dad. So what's dominant? Red. So if we put some symbols on these things, which one's recessive? White's got to be recessive, right? So the P generation female, we might write like that, X little r, X little r. The P generation male, X big R, Y. We don't do anything with a Y because what kind of trait is it? X-linked. So in the F1s, all our females in the F1s would be X big R and X little r. And all our males would be X little r and Y. And we're not going to have time to finish these problems, but one thing I will do is let's go ahead and draw the Punnett square to show what we should get in the F2. So let's draw a Punnett square between those. And if we do that, what we'll see, there's the mom gametes, here's the dad gametes. And what we're seeing is that we should get about a fourth of each possible outcome. 1/4 red-eyed males, 1/4 red-eyed females, 1/4 white-eyed males, 1/4 white-eyed females. We'll do the next week. We'll have another math day, and we will finish chi-squares and Hardy-Weinbergs and anything else we need to do.